Today's topic is, of course, cancer, and it's one of the biggest issues that we face in Australia. There are estimated to be 114,000 new cases of cancer every year. 1 in 2 Australians are diagnosed by the age of 85. More than 43,000 people are estimated to die from cancer each year. So the death rate is actually going down. So it's going down by about 16% a year. But just given our growing population and the fact that we are all ageing, more people are dying from cancer every year. The survival rates, as I said, are continuing to go up. The most common cancers at the moment, excluding non-melanomas, are prostate cancer, bowel cancer, breast and lung cancer. And of all the money we spend in research, 22% of that goes into cancer research. So that's almost just over $400 million. So we obviously are investing quite heavily. And that's why I hope today when you hear from some of our speakers, you'll get an insight into where the research is taking us in some of our key areas. So our first speaker today is Professor Andrew Biankin. Andrew is a surgeon scientist who specialises in pancreatic cancer diseases, his clinical practice is part of a specialist unit treating pancreatic disease. His research is focused on translating scientific discoveries into patient care through improved applications of current therapies, early detection and novel therapeutics. Ladies and gentlemen, Professor Andrew Biankin.
Thank you, Andrew. About 15 years ago, I retired to the lab. Hope to come out of my retirement at some stage. So what I want to touch on today is really where we're going with cancer care, cancer treatment, where things are changing, the new technologies of how they're changing the way we perceive cancer, the way we research cancer, and the way we treat cancer. It's a challenge for all of us in the community because these new technologies are not only proving to be challenging for us to adopt, but also touching on all elements of society, from the regulators, through government, through ethics committees, through the legal profession. There are so many complex interactions that need to take shape in order for us to change the paradigm of how we do this and really enact the next step in cancer care. And this is something that the Kinghorn Cancer Centre is specifically built for, and I'm not going to dwell on the Kinghorn because Allan Spigelman is going to be speaking about the Kinghorn at length. So I'll just merely touch on the fact that what we aim to achieve is to deliver the right treatment to the right patient at the right time. And I'll talk about that today, what it means, why we're uncovering these things, how we're uncovering them, and what it means for care in the future. So if you look back based on what we know now, I think we've been lucky that we've actually identified any treatments at all. We've tried to push that luck, and now we're in a bit of trouble. The pharmaceutical industry keeps adopting the same mechanisms it's used for years, and through good fortune, good planning, or good luck, that's arguable, identified some treatments. But now it's becoming more and more expensive. The reason it's becoming more and more expensive is that 3 out of 4 drugs fail in the last stages. And considering it takes about $1 to $1.5 billion to develop a drug, if 3 of them fail, it's $6 billion to develop one drug that might work in a small subgroup of people. So how we do that in the future is challenging. Integrated with that is knowledge. We acquire a lot of knowledge about science, about cancer, about diseases. But how do we push that through to changing things in patients? It was a lot easier 20, 30, 40 years ago. If you identified something new, you said, oh shit, this is good. I think I'll give it to my next patient. Everything was fine, it worked, and in fact, we wouldn't have corticosteroids today if it wasn't for the fact that a couple of guys mushed up 27,000 adrenal glands and then gave it to a woman with very advanced arthritis. They didn't do a clinical trial. They basically planned things. They gave her lolly water for 5 days, nothing changed. And then they gave her the corticosteroids, and within 3 days, she was up, walking, doing the things that she would have done normally 5 years previously, but couldn't do that because of her arthritis. If you did that today, you'd probably go to jail. So they're the challenges that we face, and in fact, if you know something and you can't do much with it, why the hell know it anyway? But we won't get existential in our discussions. So until only very recently, clinical practice and research was very integrated, particularly in Australia. But to a certain degree, we've lost our way. They're moving further and further apart. There's this cavern between basic scientific discovery and how we make a difference to patients. And really, what we're trying to achieve here is do the reverse, is start to try and break down some of those barriers. And some of those barriers are challenging because there only needs to be one particular problem and the whole thing can fall over. And so the very basis as to why we want to look at delivering the right treatment to the right patient at the right time is that we're all different. And so if we're all different, it makes sense that our cancers are going to be different, the diseases we get are going to be different. Certainly there will be some similarities, but of course there will be more and more complex differences. And so we've known for almost 1,000 years that if you're testing a new drug, you can't just test it on a mix of diseases. You want to identify that particular disease and identify that particular group. Now, if you lump cancers into one big group and say, oh, you've got cancer, we don't care where it comes from, whatever, just take this pill, it's unlikely you're going to discover anything that's going to make a difference. But as we start to understand more and more, we're starting to understand that these cancers are very, very different. They're a mix. They might look the same under the microscope, but the microscope's 300 years old. So isn't it about time we used a bit of new technology? And I'm going to talk today about a technology that's only a couple of years old. In fact, some of the technology we use is basically days old, pre-release alpha and beta testing technology. And so we're part of the International Cancer Genome Consortium. Cancer being a disease of the genes, it makes sense for us to study the underlying genetic abnormalities. Our main focus is pancreatic cancer, and we work together with IMB, in fact, where John Maddock is from, with a fellow called Sean Gribbin, who also works on ovarian cancer. You can see the incidence there of pancreatic cancer in the world, so it's an important disease, and in fact it's the 4th leading cause of cancer death. It's probably only the 10th in incidence, but if you get it, 95% chance you're not going to make it, and 50% chance you'll die within the first 6 months. 90% within the first year. So what's this new technology? This new technology is particularly suited to looking at the structure of DNA, picking all the problems with DNA in a cancer. So you compare what happens in your genes, in your chromosomes, in your cancer, compared to what the chromosomes and the genes you were born with. And you make those comparisons to identify exactly what it is in the cancer that makes it be a cancer, study that particular element and see if you can make a drug towards it. We don't do this on our own. It's together with a network called the Australian Pancreatic Cancer Network, and the project itself is called the Australian Pancreatic Cancer Genome Initiative, and it brings together leading centres across Australia. And we're trying to move towards having those leading centres not only research with us, but also start to push things into clinical practice. What sort of technology do we use? Well, these machines, you can see just in that box there, and there's a couple of arrays there. These are the new machines that basically stand on the floor. You need a fleet of them. The main difference is that you probably all remember the Human Genome Project. That took 15 years and $1.5 billion to complete 4 or 5 sequences. We can do that in a lab on its own for about $20,000 now, in about 2 days, 3 days. Cancer's a little more complex than that, so we blow a little bit more money at the moment to be able to do that, but the costs are coming down to a point where we think within the next 6 months we'll be able to offer patients some variation of genetic sequencing to look at their particular cancer and help us select the right treatment for that. And so as we study these cancers with new technology, with a better microscope, so to speak, we're starting to understand that they're a mix of diseases. So if you look on the left-hand side there, you can see breast cancer. And each one of those different bars we think is a different type of breast cancer. So those 2 big bars, we all heard about estrogen receptor positive and estrogen receptor negative breast cancer. That decides which treatment you get. A newer discovery called Herceptin, it targets specifically those cancers that have a particular genetic abnormality called HER2 amplification. But what happens when you start to uncover those cancers that happen in smaller and smaller percentages? That's very challenging. You're back to the woman with the arthritis again. I mean, can you do a clinical trial where there's only maybe 5 incidences of pancreatic cancer, say, with this particular abnormality, HER2 amplification in Australia? You'll be doing a clinical trial for 50 years. It's not going to happen. So we have to find new ways of doing that, and that's what we're trying to do. Also, it might surprise all of them. Well, it surprised me when I looked at these figures, is that everybody talks about the top 3 or 4 cancers. They only comprise about 30% of all the cancers that we see as a community. Most cancers are others, things we've often never heard of. But if you look at them as the burden to society, they probably amount to the most or the greatest burden to society. So how do we challenge those? How do we study those? How do we have treatments for them? And so this is what it all comes down to. If you're looking at a cancer, no matter how you, how you try and group it, say we're talking in this instance, we'll just call it, say, pancreatic cancer. The standard clinical trial is that you have a treatment called drug A. You think, well, you know, I think drug B is better. So you compare drug B. You do the study in 500 patients on one side, 500 patients on the other. You do some complex stats, and you go, it's not so good. It's $1 billion gone. Then you think, well, hold on a minute, I can fix this. What if I just add drug B to drug A? That's pretty clever, isn't it? So we just add drug B to drug A. We get twice the complications, but do we get twice the treatment? Twice the treatment effect? We probably don't. So theoretically, you can keep adding drugs, and you basically, you kill yourself just slightly less quickly than you kill the cancer. But what's probably really happening is that these individual patients respond differently to each cancer, to each treatment. Some of them respond to treatment A, some respond to treatment B, and some respond to both, or either. So working out which one to give first up, which can be fundamentally important, because at the moment you'll give treatment A because it works in the highest proportion. If that doesn't work, you wait 3 months, do a scan, oops, okay, it's not working. We'll try treatment B, oh, that's not working either, we'll try treatment C. In many cancers, particularly pancreatic cancer, well, that's game over. And so if your treatment for your cancer was treatment D, you want to be able to pick that first up and give it to them first up. And that's more of a challenge than you might imagine. And so what we do with our Cancer Genome Project, if it's mapped out across the patient journey on the left-hand side there, we basically take samples of fresh cancer taken out during a pancreatectomy when the cancer is removed. And then we basically throw all this heavy machinery at it. We identify all the genetic changes. And then we try and pick those that match to a drug. And when we identify those matches, we either test those in the lab or, more importantly, we try and inform those patients and enroll them in a clinical trial to say, well, we think that you've got this particular genetic abnormality in your cancer. We think this is the best drug for you. Let's match the two up. And they're the clinical trials we're moving to at the moment. For every particular cancer, we now generate a report. It's not just a report that says a one-liner that says, we think this is pancreatic cancer under the microscope, full stop, or a couple of characteristics, the cells look a bit funny. We characterize this as deeply as possible, so then we understand more about the cancer and try and infer what sort of treatment we might want to give. And this is really what the Kinghorn Cancer Centre is about, really performing those high-end personalised tests in an advanced oncology setting. And Dr. Spigelman will talk more about that later. So this is an example of when you get it right. So this is a patient who had pancreatic cancer, and if you look at this graph, this is the graph of the CA19-9 level, which basically indicates how bad the disease is. So if you look at the peak of that, this patient was 2 months before dying. He was going to die. His life expectancy was 2 months. He was identified on his genetic sequence to have a particular mutation which made him sensitive to another drug. So he was started on that drug. And you can see the CA19-9 level dropped. And if for those of you that have got really good eyesight, you can see that's 2006. And he's still alive today. And that's almost unheard of in pancreatic cancer. And so that's, rather than taking all of cancer at once, is a bite off the bits you can and identify the right drug for the right patient and give it to them first up. Central to this is a genetic pathology laboratory, not just looking at things under the microscope, but really trying to identify and develop these molecular reports that will be able to help clinicians decide which treatment is the best for that particular patient. And it becomes part of a cycle where you understand more about the patient treatment, how a patient responds. You use that to address particular questions in the laboratory. Then you do specific experiments to change that, specific clinical trials, which go straight back into the clinic. And streamlining this rather than having the enormous wall that we have now at the moment in trying to get through to clinical trials is something that we want to achieve. So we can translate discoveries far more rapidly than we have in the past. There's a strong economic argument for this testing. People say, these machines are very expensive. We can't afford to do this for each person. It might cost $1,000 a test or $2,000 a test. Considering a PET scan costs $7,000, I don't think $1,000 or $2,000 is very expensive. But even if you do say it's expensive, you make a comparison and you avoid giving that drug to those patients that aren't going to respond. This is how much you save in France, and this is courtesy of the French government, because we haven't done the analysis here in Australia. If you invest 1.7 million euros a year in this test, you will save 69 million euros in wasted drug. And you could do that for every disease, every type of cancer. And this is just the example in lung cancer. The same happens in many, many other cancers. So if I was to look to the future and see how we're trying to treat cancer in the future in the genomic age, and bear with me, this slide's a little bit complicated, but I'm hoping it'll make sense, is that the more we understand about these cancers at a molecular level, the more early detection becomes a feasibility. We detect genetic changes for early detection already when it comes to some leukemias. Similar things might be identified in other cancers. So once you're diagnosed with cancer, everybody gets a genetic sequence looking at the particular cancer. You look in the knowledge bank of experience, not just individual clinical trials, but an accumulated global knowledge bank of experience of how these genetic abnormalities relate to treatment, wherever it might be. And you use that to inform your treatment decisions. So you make a decision as to when you want to treat that particular patient. You have a backup system. You have a biomarker that you measure in the blood, and we can do that with some of our DNA measurements to see how the cancer's going, how it's responding to the treatment. And then, just like the example with corticosteroids, you give the treatment for a couple of days, you measure a response. If there's no response, you change your treatment, just like in the blue charts there. And you can see this level going up until you get treatment D, which is the right treatment, and then it responds. And then again, it's also important from a quality of life perspective to know when to give up and stop poisoning people for no apparent reason. And I'll leave you with that thought.
And I think Andrew is going off to the clinic. Am I right, Professor? Oh, there we go. So yes, thank you for coming along. What I'd like to do now is actually just talk a little bit more about the Kinghorn Cancer Centre and give you a little bit of a wander through what the building is going to look like, and then just talk about some of the more unique aspects of it, including the wellness centre that we are aiming to build. The Kinghorn Cancer Centre, the aim of it is to take the world-class research here at the Garvan and mix it with the world-class patient care that St Vincent's excels at, by bringing a focus to cancer. So the building combines both the patient care and also the researcher in the 10 floors. So physically, what does that mean? It means that those people who've been down to the chemotherapy suites in St Vincent's Hospital, they will actually pack up and move, and they will be on street level here in the Kinghorn Cancer Centre next door. And then there'll be 2 floors of clinical care. So if you're actually coming in to see your doctors or your nurses, you'll actually do that in the Kinghorn Cancer Centre. And then above that, you'll have all of the Garvan researchers. And the building itself has been designed in such a way that as you come in as a patient, you'll be able to see the researchers around you. So you all feel together on the journey as opposed to one silo and another silo. So this obviously is what the building will look like, looking down towards St Vincent's Hospital. Little Green Park Hotel on the corner, so if you do need a beer, there is always going to be one. I should explain where the name the Kinghorn comes from. So we were very lucky. The Rudd government kicked in $75 million to assist with the construction of the building, and then St Vincent's and the Garvan did a combined fundraising campaign to raise another $40 million. And thanks to the incredible generosity of Mr and Mrs Kinghorn, they put in $25 million to make the building happen earlier than we had planned. So when we do open next year, the aim is to make it fully operational. So it's an incredibly generous gift from two very passionate supporters of the work. So this is actually from street level, and so I'm going to shout. So this bit here is actually will be glass, because behind that on the other side are the people undertaking chemotherapy. But then there is this lovely vertical garden that goes up 4 stories. So there'll be trees and go all the way up to try to soften the environment from what is very harsh Victoria Street. So when you walk in, the whole building is very, it's wood, concrete and steel. So quite minimal, but it should be quite elegant with lots and lots and lots of glass. So when you walk in, this would be the main reception area. And then what you've got on every floor, so, sorry, if you go, oops. When you walk up the stairs, so there are 3 lifts, so you can walk up the stairs, or when you even come out of the lifts, there are walkways on every floor that take you to where you should be going, but the stairs don't go up straight. It's a bit like Hogwarts, Harry Potter fans. They move all the way around. So when you get up, coming out of the lifts, you can walk across the walkway, and then these, what look like wooden louvers, what happens for security at the end of the night is that those actually move across and actually lock out the entire floor. So you'll see the stairs, but not just the stairs, you've got these amazing offices that actually jut out over the central atrium, which is this big void in the middle of the building. So you can see, there's one there, and one here that sort of juts all the way out. To which my 8-year-old daughter thought that they were going to be really, really large fish tanks. Which I think is quite a novel idea. And then this is what the top 4 floors are going to look like. So the labs that are in this building are somewhat old-fashioned. So as we have developed over time, lab space has also changed. So if you go into the Victor Chang, the Pakalowi building, their labs are very similar to what we're going to have in the Kinghorn. So what we have is a write-up area. So an area where you can actually come back and do all your work on your computer, have your coffee, chat to your friends and record your data. And then on the other side is the actual lab. And then on the far section of the central stairwell and the central services are the big pieces of equipment. At the moment, what we don't have here in the Garvan, in fact what most research centres don't have in Australia, is this write-up area. They're just labs and they're rather contained. So what we're doing here is having 3 separate areas, one to write up, one to do your basic science work, and one with all the equipment at the other end. So this is just to show you, these are just computer renderings of what the building may look like. The bottom floor is supposed to go seamlessly onto Victoria Street, so quite interactive, quite open. This is what we're calling our donor alcoves. When you walk in, just to your left, there's a nice green space, a nice space of green marble, which actually has, on the big panels in the middle have the names of the people who have remembered the Kinghorn in their wills and therefore gone on to provide us an ongoing legacy, and all of our major donors are on the back wall as well. And as you can see from the renderings, lots of glass and lots of stairwell, and there's a great stairwell going sort of diagonally across the atrium. Some of those suspended offices, and again the stairs. So very open, very light, very natural. And that's what it will look like at night. So it's going to be quite a big building. You can't see the Kinghorn logo there. But there's a whole lot of facilities in it. So there's one particular room, similar to this, except that opens up and actually takes about 150 people, I think. And then there's a multidisciplinary team rooms where you can actually have all the people who are working on your particular case, as well as beaming in people via satellite, so they can all be part. So you haven't got to be in the building because one of the great things about the St Vincent and Marrow Health Network is that it goes around the country, and they're very good at doing rural and regional care. So rather than bring some of the patients in, we'll be able to beam their doctors or bring into the actual sessions so that we can do one-on-one without them actually travelling all the way into Darlinghurst. One of the more unique aspects of what the Kinghorn Cancer Centre is about is we are incorporating in the building a wellness centre. So a wellness centre is a place where, to me it sounds a bit like a day spa, but it could be. So exercise, acupuncture, nutrition and diet, massage, whole lot of mind-body therapy. So it's the first time that we've actually had a wellness centre within a cancer centre. And the reason we're doing that is that there is evidence that complementary therapies can help alleviate some of the symptoms to do with cancer. 30% of people who undergo chemotherapy stop doing it prematurely because of the stress associated. So if we can take away some of the stress that they are experiencing on their journey, we can keep them on the journey longer and hopefully increase the outcomes. What happens, we've noticed, is that a lot of patients are on complementary therapies but don't tell their doctors because there's some stigma associated with, well, I'm doing this or I'm trying that, but I don't want to tell him because it's not science, it's not relevant. And that's actually quite dangerous. So if people are doing alternative therapies when they're on particular programs, they could put themselves at risk. So by adding this into the Kinghorn Cancer Centre, it's a nice way of us adding a bit of... we of course won't be doing anything that hasn't got some scientific rigour around it, but does enable us to do some research as well, so that we can collect some data about what really does help patients who are on their particular journey. So I think that's a nice aspect of the Kinghorn Cancer Centre that I certainly wasn't expecting when I came on board. I like the fact that they were bringing the patients and the researchers together. I like the fact that they should be able to speed up developments of better care by just the simple fact that we are all in the one building. And then on top of that, the Wellness Centre, where we're really focusing on that whole aspect of the patient journey, I think is very interesting and very important. We are on track to open in the middle of next year. We will be doing some open days and some tours prior to the building opening. So I encourage you just to watch the Garvan website or through Breakthrough magazine, just to, in Breakthrough magazine, come along and have a wander around the building and see what it's like. I think it's going to be quite nice, and it does integrate very well with the Garvan. So on 3 different levels, there are breakthrough walls that go from the Kinghorn Cancer Centre into the Garvan. So there's a lot of interaction going on. And I think it'll be a great benefit to the entire precinct. So that is for July 2012. Before we go to morning tea and come back for the session with our next 3 experts, I'm going to introduce you to one of my colleagues, Carol O'Carroll, to tell you about our Partners for the Future program. Carol.
Good morning everybody and welcome to the Garvan. I must say this is a great part of my job, getting to see so many familiar faces here at the seminars and getting to know some of our supporters in the community. It's really wonderful to see you all here today. I'm sure that you will be encouraged by what you see today in the world of cancer research. It does give us a lot of hope for the future. But I think we all know that a cure can sometimes take more than 1 generation. So that's why we need to think of the future of the Garvan. And one way of doing that is by joining up with our Partners for the Future program. And this is a group of like-minded, far-reaching, far-thinking individuals who've decided to leave a gift to the Garvan in their wills. That's not my slide. I have a lovely home photo coming up. I wanted to show you my family. I'll just tell you a little bit about them anyway. Here we go. This is my husband Steve and my son Rory. And I've told Rory not to get too attached to having long curly hair. Unfortunately, my husband Steve's father has been diagnosed with prostate cancer, and he's quite old now and is living with it. And my husband Steve was diagnosed with prostate cancer also in his 40s. So he had a very aggressive form of it. But thanks to medical research, he is now in the clear. He has been treated, he's in the clear, and we're all very, very happy about that. So this is a very personal sort of request that I'm making to you today, to think of the future, because, you know, when Rory comes to a certain age, he will probably not even have to face the prospect of prostate cancer. So that gives me a great level of comfort, because you can see that many things do get handed down from generation to generation. So because of that, Steve and I have decided to leave a portion of our estate to the Garvan to think about the future of medical research. I think this is a legacy that is more than just money. It's a legacy of philanthropy, but it's also a legacy of health. Health for Rory and for all of his generation, for all of our children and grandchildren for the future. And that kind of legacy is more than money can buy. So I encourage you to think along those lines as well. I saw this quote by Dick Smith on the weekend. He was getting a little bit snippy at the Australian population, as he does from time to time. And he said in the 50s, anyone who'd done well was a philanthropist, whereas that's all changed today. I don't think it really has changed that much. And we can all become philanthropists. I know that during our lifetime, you know, we're not all Dick Smiths, and we're not perhaps able to give 25 million like the Kinghorns, but something that we can all do is leave a percentage of our estate to the Garvan to continue our work well into the future. And I love this quote also by Warren Buffett. If you've heard me talk before, you've heard this quote. And I think it just sort of sums it all up. Leave your children enough money so they can do anything, but not enough that they don't have to do anything. And, you know, I really don't have to encourage my son not to do anything. He's pretty good at that. So, you know, we're not going to leave our whole estate to him, and we want the legacy of his education and his the investment we make in him, for him to be able to make his own way in this world, but we would like to leave a legacy of health for him. So I'm sure you all believe in medical research, or you wouldn't be here today. And I personally very much believe in medical research because of, you know, the family issues that we're facing. But we have the power to alter the future DNA of generations to come. So I encourage you to consider leaving a gift in your will to the Garvan. And I'll be outside having a coffee if you'd like to come up and have a chat with me. And it's my great privilege to be the bequest officer here at the Garvan. And I've got some lovely brochures you can take away with you and have a think for when the time's right for you. Thank you.
Thanks, Carol. So we're going to break now for morning tea, only because we're going to move things around so that we can have morning tea a bit earlier, because the last part of the program is quite packed, and we're just fitting in with some of the diary availabilities of some of our key speakers. So there's morning tea is served outside. So I'm going to encourage the first 6 or 7 rows to use this door here, and maybe the back people, do you like to use the back door? So you've got to go up the stairs to that? No, just out there. Great. And then we'll just shout and call you back in a little while. Yeah, a little while. Little. What happened to give Carol a bottle? No. Sorry about that, Carol. Nicely done. Oh, thank you. You've been here slightly longer than me? No. Sorry, we'll just talk amongst yourselves for a moment.
Since February.
Since February, yes. So there we go. I'm still the new boy on the block. That's great. Dr. Goli Samimi is our next speaker. Goli joined the Garvan Research Program after completing a fellowship in the Cancer Prevention Fellowship Program at the National Cancer Institute in America. Goli's research has focused on examining mechanisms of chemotherapy resistance in ovarian cancer and identifying novel therapeutic targets in the stroma of ovarian tumours. As leader of the ovarian cancer group, Goli's research focus involves identification of methylation profiles in plasma DNA that have potential as diagnostic markers for early stage ovarian cancer. Dr. Samimi is going to give her presentation, and then she'll also take some questions from the floor if you have any. Please welcome Dr. Goli Samimi.
Thank you, Andrew, and thank you all for being here. It's really wonderful to see so many people here interested in what we're doing here at the Garvan. As Andrew mentioned, I've been studying ovarian cancer for pretty much most of my scientific career. I started in graduate school in San Diego in, 1999, so I've been in the field for a while. And what I'll do today is talk about two of our main projects that we have happening here in the ovarian cancer group here at the Garvan that are quite exciting, and then I'd be happy to answer any questions you might have afterwards. So this is just an example of the international incidence rates of ovarian cancer. And here we go. So Australia has one of the highest rates of ovarian cancer internationally, just after the US. It's about 10 cases per 100,000 women. In Australia, there's about 1,200 new cases of ovarian cancer a year, so it's a relatively rare disease compared to the types of cancers we commonly encounter, like breast cancer and colon cancer. However, where ovarian cancer is really a major problem is you have 1,200 new cases a year and 800 deaths per year. It's a very high case fatality rate. There's a few reasons for this. The main reason is that we don't have a method of early detection of ovarian cancer. So a majority of patients, about 75%, are diagnosed at late stage. And I'll show you why that's such a problem in the next couple of slides. But they're diagnosed at late stage, and late stage is defined as the cancer has spread out of the ovaries and the pelvis and is now spread throughout the peritoneal cavity and distant organs. So you can imagine it's quite difficult to remove all of the cancer by surgery. The good news is that 80% of patients do respond to chemotherapy initially. Unfortunately, most of these patients, probably 70 to 80%, will relapse within 1 to 2 years. And at that point, they'll develop chemotherapy-resistant disease. And so at that point, there really aren't many other options to treat these women. So because of this, there's really been, over the last 30 years, we have not seen an improvement in terms of overall survival in women diagnosed with ovarian cancer. So as I mentioned, there's a high case fatality rate, 1,200 new cases per year, almost 800 deaths. The 5-year survival is quite dismal overall. If you look at all the stages, it's about 30%. However, if you break this down into your stage of diagnosis, what we actually see is that women who are diagnosed early, where the tumor is confined to the ovaries, has quite a positive 5-year survival rate of 78%. But it's really these late stages where it's spread where we see this huge drop in 5-year survival. So this is another way of looking at this. In the yellow, we see the stage of diagnosis, and these are the percent of women who are diagnosed. And in the blue, we see survival. So if you look at stage 1, which is an early stage just confined to the ovaries, the 5-year survival is over 85%. The problem is just over 20% of women are diagnosed at this stage. And the reason for that is because a lot of the symptoms for ovarian cancer are very generic. It's a lot of bloating, a lot of fatigue, weight gain or weight loss, things that women we tend to experience in general anyway. So you don't necessarily get clued into the fact that there might be something wrong. Again, for stage 2, where it's spread from the ovaries but still in the pelvic region, survival rate, 5-year survival rate is 70%, but you can see how few women are diagnosed at this stage. And then we start seeing a switch in terms of stage 3 and 4, where it's spread either IP, so in kind of the stomach region, or distant metastases, so kidney or liver. You see much less survival and a much greater number of women who are diagnosed at this stage. So it's clear that we really need to focus on early detection in these women so that we can start getting them at this stage where we can not only surgically remove the cancer, but also treat them more appropriately. So really we come from two approaches here in the group. The first is looking at early detection, and this is trying to identify new biomarkers that we can use to identify women who have ovarian cancer. And this is a project that's been going on in the group for quite a number of years now, and we're just now getting it off the ground and getting some really interesting results. So I'll talk a bit about that. And then the second project, which is what I used to work on and what I brought over from my work, is trying to find new therapeutic targets. Right now, the standard treatment for ovarian cancer is surgery followed by chemotherapy. As I mentioned, the chemotherapy is not all that efficient, and most of these women end up relapsing, and so we need something else to treat these women with. So what I focus on is looking for new targets. And there we go. And where we look for new targets is in what's called the tumor microenvironment. So if this is an example, a cartoon of the ovary, you have epithelial cells that line the ovary, and they're supported by an underlying structure, which is the microenvironment. So this is a structure of cells and proteins that help kind of support the growth of the epithelial cells, the ovary cells. It provides them nutrients, it provides them food, it provides them blood, it provides them oxygen. What happens when a tumor forms, it forms out of this layer, so you get this tumor that form out of the epithelial layer, and you see an increase in a lot of factors that stimulate growth of this tumor. So whereas the normal layer may need a certain level of proteins and a certain level of factors to stimulate growth, a tumor requires much more, and so we see an increase in a lot of these factors. So now it's providing more food, more nutrients, more oxygen. So the idea is, can we target what's happening here in the microenvironment and basically, in essence, starve this tumor and stop it from growing? And that's really what my focus had been in the past and what we're continuing on here. So the way to do that is you want to, so this is an example, this is a slice of an ovarian tumor, and this dark purple is the epithelial layer that lines the ovary. And then this inside is the microenvironment. It's also called the stroma. So the idea is, can we determine which factors are expressed in this stroma, and can we target them specifically? And the approach we take is called microdissection. It's basically using a layer to specifically isolate the cells of interest. So rather than chop up this tumor and look at the tumor in general to figure out what factors are greatly expressed, we actually can dissect out the specific cells we're interested in studying and see what factors are expressed in those cells. So this is an example of what happens when we dissect out the epithelial layer, which is the tumor layer. And I don't have a picture of what we did, which was actually dissecting out the specific cells in the microenvironment. So at this point, we have these cells, and we've done some studies to figure out factors that are increased in the tumor, and now we're looking at ways to target them and see if that changes tumor growth. So that's one of the projects we're working on. And the second, as I mentioned, is to look for early detection markers. And what we focus on is alterations in the structure of DNA. So the specific alteration we're looking at is called DNA methylation. And DNA methylation is a way to change gene expression in cells. It happens quite commonly, particularly during development, and also quite commonly in cancer. So cells are turned off or turned on, and that changes the way cells grow. It changes their growth properties, which can lead to tumors. So if this is your structure of DNA, DNA has these regions in them which are called CPG islands. And these red dots are methylation. So you have a certain amount of methylation in your DNA. Some islands are methylated, some are not. What happens in cancer is you actually see an increase in methylation at some of these islands, which then turns off that gene. If a gene has too much methylation, it will be off. So we have genes in our cells that are responsible for keeping things at bay, not overgrowing, not growing out of control. That's their normal function. So in cancer, what will happen is these genes will become methylated and they'll be turned off, and that will allow these tumor cells to grow. Tumor cells, unfortunately, are quite smart. So the idea is, well, we know this happens in cancer, and we know it happens early in cancer. can we use this as a way to detect the cancer growing earlier? So what we do is we are actually we want to find a non-invasive test. In ovarian cancer, at this stage, it's diagnosed from surgery. So you suspect ovarian cancer, you have surgery, and then you're verified as having ovarian cancer, or you might just have a benign mass or something like that. Well, we don't want to have women undergo unnecessary surgery unless we're sure that they have cancer. So we want to develop a blood-based test, a non-invasive test where we can check for methylation of DNA in the blood and then get a better idea of whether or not the woman will have ovarian cancer. So what we're looking at is called free circulating DNA. And free circulating DNA is shed from the tumor and it actually circulates in your bloodstream. So if we can take blood from patients and isolate the plasma, which is just a specific fraction of blood that has no cells in it, it's just going to have the DNA. And then we can check this DNA for methylation. And what we have is we have blood from women with ovarian cancer and we have blood from healthy women who have donated blood as part of our study, just in terms of providing controls. So we can isolate the circulating DNA from the plasma of women with cancer and plasma of women without cancer and then compare these methylation patterns to determine if there are any that are specific for women with ovarian cancer. And then at that point, what we can do is then identify these patterns and apply them to a bigger study, more women, to get an idea of how well these patterns can differentiate the two. So that's the kind of the hallmark project of the ovarian cancer group here at the Garvan, and it's one that we've been working on for quite a while. The ovarian cancer group is Christina Wharton, who is a postdoctoral fellow, and she really has been the one who's been collecting the tissue and really optimizing all of our techniques to make sure we're doing this properly. Brian Gloss is a PhD student in the group who's done a lot of the methylation studies. Kim Moran-Jones is a postdoctoral fellow in the group who works on the tumor microenvironment study, and Vita Lin is a research assistant who helps all of them with their studies. All of our work is done in collaboration with Neville Hacker, who directs the Gynecologic Cancer Center at the Royal Hospital for Women in Randwick. And he's the gynecologic oncology surgeon, so we're able to work with him to collect blood and to collect tissues from women who are coming in for suspected ovarian cancer cases. And we've developed a biobank of approximately 800 or so tissues, 500 diagnosed ovarian cancer, and then 300 non-cancer cases. And so these are tissues and blood that we're using to do our studies. So we've really gotten a lot of support from Neville and from the Royal Hospital for Women. Other people who have been involved in this project, Nicola Armstrong is here at the Garvan. She does bioinformatics, so she's really smart and does a lot of the analyses that none of us are capable of doing. Pip O'Brien, who used to run this group before I did. Sue Clark and Rob Sutherland, who are both directors of programs here at the Garvan, who have been a big support. And then we also work with UNSW at the Ramchati Center, who's the group that does all of our sequencing to determine the DNA methylation. So thank you very much for your attention. I'm happy to take questions.
We have roving microphones. Jean and Marley.
Tell me what methylation is, please.
So methylation is biologically, it's basically a group that's added to a DNA sequence. So it's an addition of a structure to the DNA sequence that changes that sequence and stops the expression of that gene. And it's something that happens naturally in people. It's very, very controlled. So the main purpose of it has been through development. So genes are turned on and off depending on when you need to develop structures in your body. So whatever appendages are developing first, the genes will be on, and then as soon as it's developed, the genes will turn off. We see that happen in cancer. So the genes that regulate tumor growth are then turned off by the tumor cells, and that will allow the tumor to grow. So it's really an alteration to the DNA structure that stops a gene from being expressed. Is that clear?
There was a question down here, Carly?
now, maybe about 5 years ago, an email came through with regard to when you have your normal pap smear,
Mm-hmm.
you can ask to have this particular blood test done, which will help to show up whether in fact you do have any traces of ovarian cancer. Now, I sort of requested if I could have that, and the doctor said, well, it's not necessarily an accurate, it's not actually accurate with regard to whether, you know, you have a cancer there or not, and it can actually show a number of other problems
Right.
that may be existing, and the general attitude is that we don't go ahead with these blood tests anymore because you can subject women to a lot of unnecessary surgery. Is that true?
So I'm guessing the blood test you're talking about is a CA-125 test. Yeah, so a CA-125 is a marker. It's, as of now, the best marker we have for ovarian cancer, but it's not a good marker because it is increased in a lot of other cases. It's increased in benign gynecologic issues like endometriosis or fibroids. It's increased in other cancer types. Where CA-125 is incredibly useful is for relapse of ovarian cancer. So when I talk about the patients originally respond to chemotherapy and then relapse. So what happens is you have your surgery, you have your chemo, and at that point your CA-125 is low, 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 low. So at that point they measure your CA-125 every 3 months to see when it, if or when it starts increasing. So it's really not a test for the detection of ovarian cancer. It's really a test for whether or not your ovarian cancer comes back. And that's why what we're doing is trying to focus on detecting it before it happens or in the early stages.
Thank you.
Oh, sure.
I was interested in your first slide of the incidence of ovarian
Okay.
cancer. Could you talk a little bit about why the incidence is so high? Is it extra testing?
That's a good question. And it's, so a lot of times you'll see abnormally high incidences in certain countries where, yes, it's the matter of the fact that we can test them rather than the fact that the incidence may be equivalent across places, but because some area has better detection, you actually artificially see more cases. I don't think that's the case with ovarian cancer because we don't test for it. I mean, there's no screening process. You go in every year for pap smears or for colonoscopies or what have you, but you don't for ovarian cancer. So my guess is that it's probably genetic and environmental. It should correlate with breast cancer cases because there is a genetic factor for breast cancer that's in common with ovarian cancer. I notice also that it's quite high in China, equivalent to, and I don't have an explanation for that.
I'd like to ask, is endometriosis in some way related
To the ovarian cancer.
ovarian cancer or they call it chocolate cysts?
To this, so I'm not a physician, so I don't know how. I'm not going to be able to talk on how well these things correlate. But in general, endometriosis is not a risk factor for ovarian cancer. The only major risk factor for ovarian cancer is age. That's the biggest known risk factor. And the BRCA1 and BRCA2 gene mutations, which are big risk factors for ovarian cancer, are responsible for about 20% of ovarian cancers. But other than that, there's actually not many other risk factors that we're aware of.
Thank you. Could I just quickly ask, with age and ovarian, the ovaries that are aging, they don't always show up on the scan anyway.
Right.
Once they get very old and shriveled, they tell me. I'm wondering, should we also be worried about that happening in conjunction with ovarian?
I'm sorry, can you repeat that? Should you be concerned?
The aging of the ovaries,
Right.
and knowing that we've had a scan,
Right.
and that it has shown almost nothing there. It's very hard to find anything, I gather. That's why, because of the ageing.
Right.
Should we be worried about that process of the ageing? Does that cause ovarian cancer once they get very old?
I don't know. I wouldn't call it, it's not a cause of ovarian cancer. There is no cause of ovarian cancer. Ovarian cancer has a higher incidence in postmenopausal women. And so yes, it's presumably would be harder to detect. But it's more, I think, the idea that you have to be aware of your symptoms. As I mentioned, it's very generic symptoms. So I think the idea is to be aware of if they persist or if they get stronger or if they increase. You need to, I believe when we had a patient advocate here, she mentioned that for her the real obvious symptom was major fatigue that she could not get rid of, that persisted for weeks and weeks on end. I think when it gets to that point, it's something that you need to insist to your doctor to do more testing.
Could you please mention something about a starter to the whole process? Is there anything on that?
A starter?
A starter. What introduces cancer?
Okay.
To the beginning?
Sure. So in general, cancer comes, and this is not just for ovarian cancer, but in general, it comes from an abnormality in your DNA. So something changes. Our gene expression in our DNA is extremely regulated. Cells grow when they're supposed to grow, and they don't grow when they're not supposed to grow. And what happens is, in cells dividing and growing, they incorporate changes in the DNA. So in one case, my cell will grow and divide, and it'll mutate a gene that's normally responsible for keeping things at bay. We have a defense system in our cells where you're allowed one of those to happen and you can handle it. But once it happens twice, the cells don't know how to handle it. So that's why, for example, women who have a mutation in this BRCA gene, which is quite common in breast cancer, they have the mutation. It means they carry one abnormality. So as soon as they get a second one, then they will have breast cancer. Then they're likely to develop breast cancer, and that's why women with BRCA mutations are much more likely to develop cancer, because they only need one more abnormality. The one that they have has already been inherited. So really what it boils down to is it's a disease of our DNA not working the way it's supposed to work. And even though we do have a defense mechanism where it can happen once, it's that second time that's really detrimental. And in a lot of these the abnormalities can be inherited.
There's no outside introduction to any starter.
Well, a lot of times there could be environmental effects that might increase these abnormalities. So smoking obviously is going to increase the mutations that you have in your cells. There's some indication, which is one of the reasons why obesity has become such an epidemic, that obesity cells are going to have greater changes in terms of their DNA alteration. So clearly there is some environmental factors. I don't study the genetics of it, so other than smoking and obesity, I'm not sure what else, but it's clear that there is that indication as well. But in the end, what it boils down to is these alterations in your DNA are what's changing the way your cells are supposed to work.
Okay, last one.
Hi.
Would HRT increase the risk of ovarian cancer?
There is a link between HRT and ovarian cancer, but it's not very strong, and I don't know the details of most of these studies, but generally women who are taking an HRT is hormone replacement therapy, so that's generally postmenopausal women. So then the question is it the fact that they're postmenopausal, or is it the fact that they're taking HRT? But the link is not very strong. It's present, but it's not very strong.
Thank you, Goli. If you could thank Dr. Samimi.
Thank you very much.
Moving along, our next speaker is Professor Allan Spiegelman. Allan is Professor of Surgery at the University of New South Wales, Clinical Associate Dean at St Vincent's Clinical School and Director of Cancer Services at St Vincent's and Marrow Health Services, Sydney. He also serves on the New South Wales Medical Board and is Chair of the International Society of Gastrointestinal Hereditary Tumours. I won't be invited back, will I?
I'm sure you will. Now, I'm actually a warm-up act for Professor Tony Dodds, who's just arrived, who's going to address us about blood cancers. Professor Dodds is a haematologist and will update us as to some recent advances in that particular area. And I really wanted to speak, just in case Andrew had missed a few of the more relevant features of the Kinghorn Cancer Centre, so that I could outline those to you. But first, why the Kinghorn Cancer Centre? And all of you will know that there are several cancer centres opening up around Sydney and around the country. A huge one in Melbourne, the Peter MacCallum, $1 billion institution adjacent to the Royal Melbourne Hospital. The Lifehouse at Royal Prince Alfred, not far from here. Prince of Wales has recently obtained funding from the state government, the old state government, now the new state government, to bring together and modernise their somewhat antiquated facilities there. And they also had a generous bequest from Frank Lowy for what's called the Lowy Institute, a dedicated cancer research institute on campus. So why all of this interest? Why all of this money and funding? Are we going to be competing? Well, mostly we're going to be collaborating, but there will be healthy competition. And the reason there has to be is because although, as Professor Biankin indicated, survival for many of the tumour groups, patients with those tumours, is increasing and improving, so too is the incidence, not just in Australia but worldwide. And although St Vincent's, again Andrew indicated that we have best practice, a high reputation, and we consistently score at the top of patient satisfaction ratings for cancer around the state, that gives us no room for complacency. Because all that means is that we're as good or better than our colleagues. It doesn't mean we're doing everything we should be doing. And so when I arrived here now 5 years ago to take up the various positions that Andrew mentioned, particularly the cancer services role, we did an analysis of what we did well, what we could do better. And one of the key findings of that analysis, which became very apparent, was that there's a road between St Vincent's and the Garvan, and it might as well be the River Jordan, because the collaboration is healthy in some respects, but clearly could be very much improved. And through the mechanisms that Andrew outlined and the gracious funding through government and philanthropy, we will now very largely be working in the same physical environment. And although Andrew indicated there'll be clinical care in the first 2 or 3 floors and research in the floors above, those floors will be shared. There'll be clinician researchers who go between both areas, and there'll be researchers who will now attend our multidisciplinary team meetings where patient care is discussed, and clinicians attending research meetings. And as I said, clinicians who work in the laboratory will be able to do that work adjacent to Garvan researchers. So there's some important factors in what we hope will improve outcomes, early introduction of breakthroughs of treatment to the bedside, facilitated by working in the same building. Now, of direct relevance to staff and patients, of course, and a few things that Andrew omitted to say, there will be, and these are mundane things, but they're very important, there will be a patient drop-off area at the front of the building. There's a reason we put the chemotherapy outpatients, that's ambulatory care, people walking in and out on a daily basis for their care, on the ground floor. At the moment, it's not on the ground floor in the building across the road, and patients have to navigate the hospital. You'll be able to walk straight into that centre, be dropped off at the front, and most importantly, whoever's driving you, or if you're driving yourself, you can drive and park underneath in dedicated parking spaces for patients. That's something that we recognise as a problem for our patients in our area, and this building is going to address that. Andrew also referred to the Wellness Centre. That's an area where we think we can do better in providing better information and help on the quality of life aspect and also perhaps survival aspect for our patients, something that we're constrained in offering in our current physical environment. Equally, the clinical psychologists, social workers, genetic counsellors, all work in quite tight areas at the moment, in inadequate areas. and as indeed some of our multidisciplinary clinics are in inadequate areas. That will all be addressed in the new building, and that's going to be very important for provision of up-to-date care. The counsellors, social workers, psychologists I mentioned will have dedicated interview rooms. They won't be, as they are now, clinically designed rooms. They'll be rooms that will give patients an environment in which to relax, as will the wellness centre, which is purposely designed as to have a day spa feel. So we're trying to get away from that harsh clinical environment. And we've also divorced the administration of chemotherapy from where patients are seen for consultations and for social work, etc. Because the feedback, not just here but from around the world, is that some people experience anticipatory nausea if they have to go back to the same area where they had their treatment to get further follow-up. So all those things we will address in the new centre. Finally, penultimately, Andrew indicated that we provide rural and regional services to New South Wales, and Tony Dodds' team, they do that for haematology oncology for a number of centres outside of Sydney. It's part of our mission at St Vincent's. I myself run the hereditary cancer clinic at St Vincent's Hospital, and as a result of that provide outreach cancer genetic services. That's the sort of breast cancer gene testing we've heard about, bowel cancer gene testing for eligible people with a view to early detection and preferably prevention, which we can achieve for many people at risk who are shown to carry these gene mutations. And the areas that we provide that service here at St Vincent's range from Gosford right up to the Queensland border. So yesterday, for example, I was in Port Macquarie. In a few weeks I'll be in Coffs Harbour, Tweed Heads, Kingscliff, Lismore. The list goes on. We also provide the same service to Goulburn, Queanbeyan, Merimbula, Maria on the south coast, Newcastle, Tamworth, Dubbo, Taree. All of that coordinated through St Vincent's. If we didn't provide that service, those patients who we regard as actually underprivileged because they don't have the privilege of access that we do in the major metropolitan areas would not have access or would find it very difficult. And we increasingly move towards telehealth in providing that service. The other and the final thing to say before handing over to Tony is that perhaps most importantly to recognise that Tony heads our bone marrow transplant service. That's a state-based service again, providing a service for patients across the state. And in the basement of the new building, apart from parking, will be a bone marrow transplant best practice facility, which we desperately require to enable us to continue to function in that role. And so without further ado, I'm going to ask Tony to come to the podium.
Thank you. I'll just open up this. Okay, so good. Thank you, Allan. Thank you very much. I think I've gone one slide ahead, so I'll just go back. Today, thank you. Allan mentioned our transplant unit, which we're very proud of, but I'm not really going to talk about transplants today. I thought I'd maybe focus on something a little bit novel where we see the future of haematology, because as many of you know, maybe some of you have been treated for cancer or even some of these blood diseases, you'll know that the current treatments are very unsatisfactory with intensive chemotherapy, which has a lot of side effects, both short-term and long-term. And they're obviously things that we don't get pleasure out of either, so we're anxiously looking to the future in terms of new sorts of therapy. So what I'm going to talk about today is what we call targeted therapy, where we're hoping that we'll be able to aim that therapy particularly at the tumour and avoid a lot of the undesirable effects associated with chemotherapy. So to understand blood diseases, I guess you need to understand a little bit about where they come from. And there's this guy here called the Hemopoietic stem cell, and this is the fellow that we transplant when we do a bone marrow transplant. And we're able to repopulate someone's body with their whole immune system, the lymphoid cells, as well as the myeloid cells. But this is a useful model to look at cancers, because the cancers of blood derive from one of these cells, and that's how we categorise and define them. So if we look at broadly what sorts of blood cancers we have to deal with, the one I guess people fear most is leukemia, and that could be acute leukemia or chronic leukemia. There's a condition called myelodysplasia, which unfortunately is becoming very much more common now as our population ages. Myeloproliferative diseases, where we have too many blood cells, lymphoma and multiple myeloma. Obviously, I'm not going to cover all of those in the 10 minutes that I've got. But I wanted to focus on targeted therapy. I'm not quite sure what people have said before this, but this, I guess, is more specific about blood cancer cells, because these are the ways that we can directly attack that blood cancer cell without attacking, hopefully, the normal cells, which is the current problem that we have. So here's our cancer cell, which has lots of things inside it which make it multiply and behave like a cancer cell. There are ways that we can specifically attack that. One of those is immunotherapy, and that's really what a bone marrow transplant is. It's an immune attack using someone else's immune system on that cancer cell, but of course it has undesirable side effects as well. We have currently in use now therapeutic antibodies which specifically attack an epitope or an antigen on that cancer cell, and there are going to be more of these in clinical trial. In fact, we're involved in clinical trials at the moment. We can use this tyrosine kinase enzyme. We have direct inhibitors. I'm going to briefly give you an example of that. There are signalling molecules. There are lots of different ways that we can attack this tumour cell. I'm just going to tell you about a couple of them today. Okay, so this is a little bit complicated, but when we think about myeloproliferative diseases, we've now already identified specific mutations in some of these types of diseases that we can attack. And one of these diseases is called chronic myeloid leukemia, which was not so long ago a terribly unpleasant disease which was universally fatal within an average time of about 5 years. Now with a new therapy, more than 90% of people are virtually cured of that disease. So we're going to focus on that one first. Now here's a picture of what chronic myeloid leukemia looks like under the microscope. Way too many of these immature white cells in the blood. And what happens here is that there's a swapping of material in the chromosomes. So we have chromosome 9, chromosome 22, and bits of this swap over for a reason we don't understand to produce a new gene, a novel chimeric gene, which produces a new protein, and this protein drives the multiplication, the cancer behaviour of that cell. And that's only really been discovered in the last 5 years. So what was discovered when we realised this was that there was a specific inhibitor which could inhibit that enzyme which is driving the cancer. That drug is called imatinib or Glyvec, and many of you would have perhaps heard of that in the media. So here's a bit of a cartoon of what's happening here. Here is the chromosome, the translocation there, the abnormal gene that's being formed in this cancer. This is the fusion protein, the new protein, which isn't normally in our blood cells that's formed, and that drives this disease of CML. So what if we use something magic? Here's the protein again. We have this drug, imatinib, and there is now 3 or 4 other drugs which are even more potent than this. It blocks that protein and therefore turns off the hemopoiesis. And that's a tablet. It's not a chemotherapy drug. It's a tablet you have to take perhaps for the rest of your life, but very few side effects, and a magic treatment, the first of these targeted therapies I think that we had available to us. Okay, what about another disease? You may have heard of polycythemia, where you have too many blood cells, or thrombocythemia, you have too many platelets. It's a chronic disease, but ultimately it can turn into leukemia, so we're anxious to try and block that. And there's in fact been another mutation. This was really only described about 4 or 5 years ago in the New England Journal of Medicine, and this is a mutation which is called a gain of function mutation. What does that mean? It means it turns on the whole process. It doesn't stop it. The mutation drives more and more production. The same sort of problem drives a clone of cells to keep multiplying and avoid control that normally happens in the bone marrow. Now this is a little bit complicated, but just to simplify it to you, there's this pathway called the JAK-STAT pathway, which is a controlling pathway in blood cells. So when you have a ligand, say erythropoietin, that binds onto the receptor, it tells the bone marrow to make more of these cells. So what if you've got a mutation here which turns the whole thing on the whole time? It's like the Paul Keating expression, the switch is turned to positive rather than negative. So when the switch is turned to positive, this keeps being driven on, and therefore you accumulate more and more cells, as what happens in polycythemia. So now already there are being inhibitors of this JAK2 mutation. These trials, we're actually involved in several of these trials right at this moment. The drugs are not even available yet. They're subject to clinical trials. And these inhibitors of that mutation already decrease spleen size, already improve quality of life, already reduce the blood count. So they seem to be successful. It's another tablet. Rather than a chemotherapy treatment, it's a tablet one can take to turn off that process simply by recognising what is the genetic change, what is the mutation that's occurring. Okay, so I'm going to turn now to a type of leukemia. This is called chronic lymphocytic leukemia. It's the commonest leukemia that affects Western populations. It results in a whole lot of lymphocytes occurring in the blood and enlargement of the lymph glands, generally all over the body. Now this is very, very topical because only last week, and this received media attention, there were cells developed in the United States in which not to use the technical word, but the cells were re-educated. So the person's own lymphocytes were taken from the body, the normal lymphocytes, the T lymphocytes, and re-educated to attack the leukemic cells in this process. And this is one of the reasons we may be able to use the new lab in the Kinghorn Centre to do this sort of work in a clean stem cell laboratory. So these CAR cells, or modified T cells, resulted in attacks on the chronic lymphocytic leukemic cells, and in one patient a complete remission was obtained by that process. So this was really only a pilot trial in which autologous, the person's own T cells expressing a particular antigen, were used, and that was made in the laboratory, and those cells were then used to attack the CLL cells. And 3 patients have been described, of whom 1 obtained a complete remission using that process. So here's just some pictures of how that happened. Here's the person's bone marrow with a whole lot of abnormal cells in the bone marrow. By day 23, a lot of those cells were gone, and by 6 months, the bone marrow had returned completely to normal. So this is using the person's own cells by re-educating their own immune cells to attack the leukemic cells. Here's just some X-rays, some CT scans with enlarged lymph glands, which also gradually disappeared. So you can see the future is very exciting for us, and we'll be very excited to be over here in the new Kinghorn as partners with the Garvan Institute to do some of this more basic research, to be able to develop more of these sort of attacks on the leukemic cells rather than attacks on the normal cells. So just to finish off, this is a 35th anniversary celebration we had last year. We've been doing bone marrow transplants for 35 years, and we've done over 1,000 of them, and here are a whole lot of transplant survivors now from that process. So we're not going to give this up, obviously, but we're very keen also to look at newer therapies and keep the whole area moving forward. So thank you very much.